All right, good morning, everyone. My name is David Romero. I'm a PhD student at the Cousins Lab, ID2, and I'll be chairing this morning's session. So, once again, good morning, everyone, and I'd like to welcome you to this morning's postgraduate seminar. Before we start, I'd like to acknowledge the traditional owners and custodians of the land which we meet today, the Wurundjeri people of the Kulin Nation. I recognize that this land where we have the privilege to work, study, and play is unceded, and yet is still taken care of by the elders of the community, past, present, and emerging. They were the first teachers and scientists in Victoria, and just as we are here to learn from the seminar today, we too have much still to learn from the elders of the community. Okay, so once again, I welcome everyone to today's seminar entitled Association Studies, that's my script is wrong, Genetics of Complex Traits, Genome-Wide Association Studies, and Polygenetic Risk Scores to be led by Dr. Vicki Jackson. So Dr. Vicki is a senior postdoctoral researcher and a statistical geneticist in the Population Health and Immunity Division. Vicki completed a master's in medical statistics and a PhD in genetic epidemiology at the University of Leicester in the UK before moving to WEHI in 2017. Her research focuses on understanding the genetic basis of a wide range of human traits and diseases through statistical analysis of large-scale genetic and biologic data. Vicki's current work spans both complex traits and rare diseases, with a primary focus on investigating the genetic determinants of speech disorders, retinal and hematological traits, and cancers. Her previous work focused on genetics of respiratory health and smoking behavior. Vicki has extensive experience of leading analyses of large genetic datasets and expertise in developing pipelines for undertaking genome-wide association studies, or GWAS, phylogenetic risk scores, and other statistical genetic methods. So without further ado, I give you Dr. Vicki.
Thank you, David, for that introduction. So, cool. I'll dive straight in. So this is just a quick overview of what I'll be talking about today. So first of all, I'll go through some key concepts that are relevant for genome-wide association studies, which are also known as GWAS. I'll give a quick overview of how to perform a GWAS, followed by a kind of short history of GWAS studies. And then finally, I will talk about how GWAS are being turned into polygenic risk scores and how these may have some clinical utility. So first of all, some relevant key concepts. So what is a genome-wide association study? So this is an experimental design that's used to detect associations between genetic variants and traits in samples from populations. So breaking that down, the sort of genetic variants that we're interested in are primarily single nucleotide polymorphisms. So these are just single base pair changes in the genome. We might also look at some small insertions and deletions, but primarily SNPs. The traits that we're interested in are complex traits. So what a complex trait is something that's influenced by a range of genetic factors in addition to non-genetic factors, so things like the environment, lifestyle, and so on. And complex traits could take the form of a quantitative trait, so something you measure, like your height or blood pressure, something like that, or it could be a disease. And GWAS is generally undertaken in sort of population-based studies, and that's opposed to the family-based studies that Halim spoke about last week. So this plot here just sort of summarizes genetic architecture of disease. Last week's talk from Halim focused up in this corner here. So that's very rare genetic variants that have a really high penetrance and are largely causal for disease. With GWAS, we're more focused in this corner down here. So it's generally variants that are common within a population which individually have quite small effect on disease. But increasingly, as sample sizes for GWAS get bigger, we're looking more in this middle area here of lower frequency variants with more intermediate effects. So GWAS was sort of really made possible through this international HapMap project. This is an international consortium that aimed to determine variation in individuals with African, Asian, and European ancestries. So this project aimed to determine where in the genome common variation existed. So they sort of found common SNPs across the individuals in this project. And then they could generate haplotypes for these individuals, that is, sets of variants that tend to be inherited together. Now, as these SNPs tend to be inherited together, we find that within the population, the alleles of these SNPs are correlated. And this correlation of SNPs across the genome is known as linkage disequilibrium, or LD, and this is really key for the way GWAS work. So as these SNPs were inherited together, they were able to identify a set of tag SNPs which kind of represent these haplotypes and capture a large proportion of the common variation genome-wide. So given we had this set of tag SNPs, this sort of led to the development of genotyping arrays which measure these tag SNPs and basically allow us to study common genetic variants in large numbers of individuals. Not moving. There we go. Okay, so when we've measured genome-wide variation in our population, the next thing we want to do is look for associations between our measured SNPs and traits. So in this example here, for a quantitative trait, we've got a SNP of interest, which has two alleles, the A and the G. And so there's three possible genotypes, AA, the heterozygote GA, and then the homozygote GG. And you can see in this example, the GG genotype is associated with higher values of the trait compared to the AA genotype, and the GA is somewhere in the middle. We can also do this for a disease. So in this example, you can see that those with the GG genotype are enriched for the disease compared to those with the AA. So these sorts of SNP trait associations are what we're interested in. Now, in kind of traditional epidemiology, that's something we do, look for associations between two traits. So you might, for example, observe an association between depression and poor sleep. What you might be really interested in, however, is whether depression causes poor sleep. So this is really difficult in a kind of observational study because there's several explanations for why you might see an association between traits. So one could be there might be a sort of third variable, which could be related to lifestyle factors. So the amount of physical activity or the amount of alcohol you drink, these could be related to both depression and also they could relate to poor sleep. And in this case, you would sort of see an association between depression and poor sleep, but this was due to confounding of these lifestyle factors. Alternatively, it could be that if you have poor sleep, this could then lead to development of depression, and this is known as reverse causation. So do these things also occur when we're looking at SNP trait associations? So say we're looking at a SNP to a disease association, reverse causation is not going to be possible in this case. Having the disease is not going to alter your DNA. Similarly, confounding due to kind of lifestyle factors or other environmental factors is not going to occur for the same reason. However, confounding is possible in these sort of SNP associations, and that's due to population structure. So what's meant by this? So population structure is an issue where, first of all, your study population has different subpopulations within it, which have different SNP allele frequencies, and that's going to be due to different genetic ancestries of the people in your study. The second thing is where between these different subpopulations, the trait varies in some way. So that might be different trait distributions or different prevalence of disease in these two subpopulations. And in this way, population structure can act as a confounder. So population structure can affect the trait through several mechanisms. So it could be due to different environmental exposures between the subpopulations. It could be different socioeconomic factors or even cultural factors. So I'll just go through an example to explain how this can cause associations to appear where they don't exist. So we've got this population. Here, red represents an individual having the disease and purple no disease. And the circle indicates they have a SNP and the square no SNP. We just assume presence-absence in the SNP for this example for simplicity. So in this population, the SNP has a frequency of 0.8. The probability of disease in those who have the SNP is 0.2, and the probability of those who don't have the SNP is also 0.2. So you can see there's clearly no association there between the SNP and disease in this case. Now, let's say we have a second population. In this population, the SNP occurs at a lower frequency, but the disease is more common. So SNP has a frequency of 0.4 now. The probability of disease for individuals who have the SNP is 0.6, and for those who don't have the SNP is also 0.6. So again, there's clearly no association between SNP and disease. Now, let's suppose we have a third population, which is made up from a combination of population 1 and population 2. In this third population, the frequency of the SNP is 0.6. For those who have the SNP, the probability of disease is about 1 in 3. And for those who don't have the SNP, the probability of disease is higher at 0.5. So here we see that the SNP appears to be associated with a lower risk of disease. And we see that that association just comes about due to the structure of the population made up of these two different subpopulations. So luckily, population structure is something that we can capture through principal component analysis, or PCA. So what PCA is a form of dimensionality reduction. So it transforms the really high dimensional genetic data to a small number of variables known as principal components. And these principal components still capture much of the original variation captured by all the measured SNPs genome-wide. And this is a useful tool for population structure identification and can help reveal similarities and differences between populations and allow us to visualize population structure and ancestry. So this is an example of principal component analyses carried out as part of the Human Genome Diversity Project. So what these plots show on the left is PC1 versus PC2, and then on the right, PC3 versus PC4. And each dot here is an individual, and they're coloured by their ancestry here. And what you can sort of clearly see is that individuals from the same ancestry sort of tend to cluster together in this PC space. And you can clearly see different clusters emerging. So this isn't just the case on sort of a global level. Population structure also can exist within continental ancestries. So this is a PC analysis of just individuals with European ancestries, here coloured by the countries corresponding on the map. And you can see quite well that PC1 is capturing north-south variation, and PC2 is capturing east-west variation, and the PC plot actually resembles the map of the countries to an extent. Okay. So, the next part of this talk, I'll just give a quick overview of how to perform a GWAS. So this is an example of a GWAS workflow. So generally, when you undertake a GWAS, this involves quite a lot of quality control of your data, usually followed by imputation, a little more QC, followed by association analyses, and then you identify your significant SNPs and genetic loci, and carry out some SNP to gene mapping. So I won't go into all of these steps in too much detail. I'll just give a quick overview. So for QC, this is generally carried out firstly on a sample basis, where you're aiming to identify individuals who've got poor quality data, which might be due to things like sample contamination, or potentially sample mix-ups. For example, if their genetic sex or relationships don't correspond with what you would expect, then you do some QC on a marker or SNP basis. So you identify variants with high missingness to exclude, and variants where the overall frequency or the distribution of genotypes are not looking quite right as you would expect. And then finally, there's often things like batch effects that we need to investigate, maybe remove SNPs that are subject to batch effects, and so on. So after QC of the data is imputation step. So your measured genetic data probably will consist of somewhere around 600,000 to 1 million SNPs, which is a lot, but by no means capturing all variation genome-wide. So what you want to do to get more SNP information is impute. And what you do this using a reference set of haplotypes. So for example, from HapMap, or there's several other reference databases out there. So you take your measured genotypes and you compare your data to these reference set of haplotypes, and you phase the data, so you work out the haplotypes for your individuals, and then you basically compare your haplotypes with the corresponding reference haplotypes and basically fill in the gaps. So in this way, you might start off with a set of 600,000 measured variants, and after imputation, you may have more like 10 million variants on which to perform your GWAS. So the association analysis itself is fairly straightforward. So in the case of a disease, you're doing a case control study. You do this using a logistic regression for each SNP. So your outcome is your disease status phenotype. And in your model, you have a term for the SNP. So your genotype will take the value either 0, 1, or 2, corresponding to the number of copies of the affect allele an individual has at the SNP. And then you will also include some additional covariates, and these include your ancestry principal components, but they could also include things like sex and age, and other covariates that are specific to your trait. There are some more complicated models. So, for example, if you have related individuals in your GWAS, there are more complicated models which can account for that, but this is sort of the most basic association test. And so that's pretty much it, but you just have to run this several million times for the several million SNPs in your GWAS. And then to kind of illustrate what the results of a GWAS look like, I'll give an example of a GWAS that I've recently been involved in, which is of retinal thickness in the UK Biobank. So first of all, a bit of background on the retina. So this is the light-sensitive area of neural tissue that can be found at the back of the eye. So the retina is comprised of several layers of really highly specialized cells which capture the light, and they then convert this to electrical signals, which is then transferred to the brain via the optic nerve, and that's what ultimately allows us to see. So the thickness of the retina is an important phenotype, and thinning or thickening of the retina occurs in several retinal diseases. And retinal thickness is a general biomarker for retinal health and has been proposed as a marker for some neurodegenerative diseases as well. So the UK Biobank is a very large study of 500,000 individuals from the UK. So these are older individuals who were recruited between 2006 to 2010. They were all aged around 40 to 65 at the time of recruitment, and they've been followed up since. And there is a wealth of data available for these individuals. So they all have filled in several questionnaires. They've had a number of physical measurements taken. Biospecimens have been collected, imaging data, and so on. And as part of this, they've all got genome-wide genotyping. And one of the imaging phenotypes they've got is from optical coherence tomography, or OCT imaging. So this is a non-invasive imaging technique that uses light waves to capture cross-sectional images of the retinal layers. And these cross-sectional images are sort of combined to sort of build up this map of the retina. It sort of looks like this. It looks a bit like a volcano. The sort of center bit here is called the fovea, and it's thinnest at this part, and then it sort of increases in thickness before going down again. And this sort of area of the retina is about 6 by 6 millimeters, so really very small. So this imaging is available in a subset of UK Biobank, about 50,000 individuals. So we've got these OCT images. They've been reprocessed to give this really high density map of thickness. So this sort of map, if you like, is a grid of 128 by 256 pixels. So there's about 29,000 pixels in total where we've got these retinal thickness measurements. So we've carried out GWAS on these thickness measurements with the aim of, first of all, identifying SNPs and genetic regions that are associated with retinal thickness. And secondly, because we've got this really high resolution phenotypes, this should enable us to look at spatial patterns of these associations within the retina. So this here is a Manhattan plot for 1 pixel. So GWAS studies are usually presented in this form. So what you're looking at here is each dot represents a SNP, and they're all ordered by chromosome and position. And on the y-axis is the minus log 10 p-value. So basically the higher up the dot, the more significant the SNP is. And you will see that a lot of these, they kind of have towers of association, and this comes from the linkage disequilibrium and the way that the SNPs are inherited together. So you don't just get one significant SNP, you get a lot of SNPs within a region that show association because they're all highly correlated. So we undertook these GWAS for all the pixels, and basically across the whole retinal grid, there were 224 genetic associations identified in total. Now, you can see On these plots, this red line here represents genome-wide significance level, which is a p-value of 5 × 10⁻⁸. And generally in GWAS studies, anything that kind of comes above this level is reported as genome-wide significant. Now you can see there's obviously a lot of SNPs that fall above this level, but due to the LD, many of these are correlated. So what we do when we present our results is we sort of select one, which is usually the most significant SNP, to represent our independent genetic regions. And the results then might be presented in a table like this. So this is our top 10 associations. They're associated with all different pixels here. And in these results, you've got information about the SNP, so the chromosome, the position, and then the SNP ID, which is usually this RSID number here. And then you've got information about the alleles. So you have the affect allele and the non-affect allele. The affect allele frequency, so that's just the frequency of our affect allele here in the population. So you'll see that all of these have frequencies sort of 7% and higher, so they're all pretty common in the population. And then the beta here is in respect to the affect allele. So on this top row, the G allele here, or for each copy of the G allele, that's associated with an increase in your retinal thickness of this pixel of about 2 units. There's the standard error of the beta, and then our p-value here, which you can see are very small. These are all very highly significant. So once we've identified our SNPs, the next thing we want to do is to link our SNPs to genes. So we do this by bringing in various other data sets and pieces of information. So in this case here, we first of all asked whether the SNP was located within a gene. If the SNP is within a gene, is the SNP predicted to be damaging? So does it influence the resulting protein of the gene? We asked whether the SNP is associated with expression of any genes in retina, brain, or blood. And there are many data sets out there. They're called EQTL data sets, or expression quantitative loci, which have for various different tissues, and they're effectively GWAS of the expression of multiple genes in that tissues. So you can overlap your GWAS results with the EQTL results to determine whether it's likely the SNP you've identified is influencing gene expression. And finally, we asked whether the SNP overlapped a chromatin interaction site in retina or brain. And this along the bottom just shows some of the top hits and how each of those were mapped to genes. So I'll just give a couple of examples for some of our top hits. So this first SNP here, the plot on the left shows the association results across that grid, so across all pixels. On the left shows the beta, so the effect of this SNP on each pixel. So the blue is associated with thickening of the retina, and the red associated with thinning. On the right-hand side is the minus log 10 p-value. So the darker the color, the more statistically significant the association. So this particular SNP, you can see that the association is really focused in the middle bit. This bit's known as the fovea, and it's associated with thickening of this part. So this SNP, using the EQTL data, we link the SNP to this gene T-span11. So this SNP looks like it influences expression of this gene in both brain and retina. And this SNP has also come up in other GWAS, so it's for optic disc morphology, and so another eye-related trait. This second SNP example here shows quite a different pattern of association. So the association is strongest in this sort of bottom corner, so this is towards the side of the nose. So this SNP was actually located within a gene, FOXP1. And again, this SNP has come up in other GWAS, so for intraocular pressure and vertical cup disc ratio, and both of these are related to glaucoma. This gene in mice has been found to regulate the timing of early retinal cell production. So a few relevant things there. So once we have mapped our SNPs to genes, what we then might want to do is look at whether our implicated genes show any enrichment for any biological pathways. So we've done this by overrepresentation analysis of gene ontology terms. And what comes up is, I guess, somewhat expected and reassuring, largely things related to eye development, vision, and so on, also cell differentiation. So things that you would perhaps expect that suggests that the genes we're picking up are relevant for our retinal phenotype. Okay, so I'll now just give a kind of short history of genome-wide association studies in general and how they've taken off over the years. So first of all, this is the first genome-wide association study that was carried out back in 2005. This study was for another eye-related disease, age-related macular degeneration. And this GWAS included just 96 cases and 50 controls, and they looked at 116 SNPs. And they found a significant association in the complement factor H gene, or the CFH gene. And this association has actually subsequently shown up in AMD GWAS consistently and did turn out to be a true association. So a couple of years after this came out this study from the Wellcome Trust Case Control Consortium. So this study looked at 7 major diseases, common diseases, and for each disease they took a set of 2,000 cases and they compared them to a set of 3,000 controls. And they found across the 7 diseases 24 different association signals. And this study was important because through it many of the methods for data quality control and the methods for association analysis were sort of developed, and a lot of the principles developed in this study are still in use today. And you can see the plot for this study. These are what we recognize as Manhattan plots. To show you how GWAS have kind of progressed, I'll just give the example of height. So height is a phenotype that's obviously very easy to measure. It's very easy to self-report, so it's collected on very large numbers of people. So in 2011, the Giant Consortium undertook a GWAS which included 183,000 individuals, and they identified 180 associations. In 2014, 250,000 individuals were in their GWAS, and they found 697 associations. In 2018, they were up to 700,000 individuals, 3,290 associations. And then more recently, in 2022, they had a GWAS of 5.4 million individuals, and they found 12,000 associations. And this is, as far as I'm aware, the largest GWAS carried out. And they presented their results like this, so they've kind of done away with the Manhattan plot. That probably wouldn't be informative. So what this is a Brisbane plot, because the first author was from Brisbane. And what this shows is the density of signals in regions across the genome. And this sort of story for height is similar for many traits. So this plot here just shows across the years, in blue, the number of GWAS-related publications there have been. In red, the average sample size of these GWAS. In green, the number of loci identified from these GWAS. And you can see it has really increased. In 2017, you might note there's a bit of a kind of uptick And this corresponds with when the UK Biobank, which I spoke about earlier, released their genetic data on 500,000 individuals. And they made this available to sort of the whole research community. So everybody suddenly had 500,000 individuals to do a GWAS on. So that kind of explains that little boost there. So what's driven the success of GWAS? So in contrast to the candidate gene studies of the past, GWAS are generally large or very, very large, and therefore well-powered in terms of the statistics. They can take into account ancestry and relatedness, and they don't rely on existing biological knowledge. Instead, GWAS sort of use a hypothesis-free approach to identify gene-trait associations. And the results from GWAS have been found to be highly replicable. Like that first AMD locus has been found in every single GWAS of AMD since. There's some areas of caution, however. So many of the SNPs and the loci identified through GWAS are not actually within genes. So therefore, the part where you map the SNPs to genes, that can be a bit tricky, and making sense of your GWAS results can be a little difficult. Another major limitation is that to date, GWAS have been very European focused. So a lot of the large studies that have genetic data available, like UK Biobank, are largely in sort of UK, Europe, and the US, and they're generally heavily biased towards individuals with European ancestries. So that is changing. There are many biobanks emerging, particularly in Asia, and more focused on studies of people from different diverse ancestries. And this is important because the associations that we see, they don't necessarily transfer across different ancestral populations. And then confounding is still an issue in GWAS. So due to population structure, but then there's also some other things which I haven't been through today, like assortative mating and indirect genetic effects. And they can confound your GWAS and make interpretation difficult. So in terms of what's the use of GWAS, one utility is that it highlights biology, which can be at times unexpected. It's proposed to aid drug target identification. So if your GWAS are sort of highlighting a number of genes in a particular pathway, and there is a drug that targets that pathway, then that drug could potentially be useful for your disease. And finally, prediction is also starting to become possible for some traits through the development of polygenic risk scores. And this leads into the last part of the talk. Okay, so polygenic risk scores. Now, individually, the SNPs that we identify through GWAS, they generally have pretty small effects on a trait or risk of disease. However, if we take all those SNPs together, we could aggregate them into what's known as a polygenic risk score. And this is proposed as a more useful predictor for disease risk or likelihood of a trait. So how do we construct these scores? They're simply a weighted sum of your SNPs across the genome. So for each individual, you have a set of SNP measured. You've got the dosage of the SNP, so that will take the form of a 0, 1, or 2, depending how many copies of the allele you've got. And then each SNP is weighted, and these weights come from the GWAS. So these are the betas, the effect sizes of each SNP. So you sum the effects of all these SNPs together. Now, one question is to do with, which variants from the GWAS do we select? So obviously you have a Manhattan plot that looks like this, and as we said before, there's a lot of correlation amongst these SNPs. So there's several methods for selecting the SNPs that go into a polygenic score, but what they all do is sort of take into account the LD structure, and they aim to select SNPs which are kind of independently associated with your trait. Now, also, schools often don't just select SNPs which meet this genome-wide significance threshold. They often take SNPs below this threshold as well. So if you construct this score for a set of individuals, you'll find that they follow broadly a normal distribution. And if you've got a set of cases for a disease and a set of controls, what you might see is a sort of difference in those two distributions of the scores between the cases and controls. And the sort of differences in these distributions is generally quite small. It might look like this. So the majority of people would fall kind of in the middle of the distribution here. And for these individuals, the polygenic score might not tell us too much about their risk of disease. But where it starts to matter is more in the kind of extremes of the distribution here. So can these scores be predictive of disease? So this study looked at coronary artery disease in UK Biobank. So they had this PRS for coronary artery disease, and they found that those in the top 8% of the distribution had 3 times odds of having a diagnosis of CAD compared to the individuals in the lowest 92%. For those in the top 2.3%, the odds were 4 times, and for those in the top 0.5%, the odds were 5 times. So another way they looked at this was to look at the prevalence of CAD in each percentile of the polygenic score. And in the top 1%, more than 10% of these individuals had a CAD diagnosis, compared to those in the bottom percentile, which was around 1%. And what this study found was that individuals in the top end of the distribution actually had similar increased risk of CAD as individuals who had pathogenic variants in known familial hypercholesterolemia genes. So where you're at these very extreme ends of the score distribution, there's potentially, these are of clinical importance. So the final example I'll give is of breast cancer, and this is one disease where polygenic scores are starting to be actually incorporated into the clinic. So breast cancer is the most common cancer diagnosed amongst women worldwide and is associated with significant mortality. So there's a number of risk factors. Number one is there are several known highly pathogenic rare variants in things like the BRCA genes, which cause breast cancer, having a family history, lifestyle, hormonal and reproductive risk factors, mammographic density, and there's also demographic factors. And common genetic variation also plays a role in breast cancer risk. So there's a number of risk prediction models for breast cancer which have been around for years and are used in clinical practice. And more recently, some of these have actually started to include polygenic scores in their risk prediction models. So one of these prediction models is called BODICEA, and this has been around for a few years, which included those sort of standard risk factors, family history, and so on. It's also included those rare, highly pathogenic variants. And this study here shows what the addition of the polygenic score to that model offers. So they validated this risk model in a prospective cohort of 66,000 women. Now looking on their own at the polygenic score, family history, mammographic density, and traditional factors, all of those on their own showed some predictive ability for breast cancer based on the area under the curve, the AUC. So these plots here just show the predicted risk on the x-axis versus the observed risk on the y. As you can see, they're all pretty correlated. But across all of the models they looked at, the best ones were those that incorporated all factors, including the polygenic scores. And this was the case in women with and without a rare pathogenic variant. So what does this look like in terms of risk? So without these models, a woman with no family history will have a lifetime risk of breast cancer of around 12%. And that's represented by this kind of dark red line here. What these risk models do is they allow a sort of more personalized estimate based on distributions of risk. So this plot shows what these distributions look like, and there are slightly different versions of the risk model here. And this blue one here includes the traditional risk factors, mammographic density, and the polygenic score. So with these distributions, a woman might be given a risk that could range from around 3% up to over 35%. And from this estimate, they could be classified as having normal, moderate, or high risk of developing cancer by age 80. And then there's a similar for individuals for women who may have a family history or a known pathogenic variant. And these sort of distributions mean that some women even with a family history or pathogenic variant may actually have just a sort of normal risk of developing breast cancer, whereas others may have a lifetime risk that exceeds 50%. And these women may benefit from additional screening or other preventative treatments. So just to summarize polygenic risk scores, these are sort of proving to be quite a useful tool on a population level. In terms of clinical utility, where they should be most useful is where they're considered as a risk factor in combination generally with other traditional risk factors. And they have the potential to improve things like screening or treatment options for some individuals. Again, there's many limitations with them. So on their own, it depends on the trait, but they might not always be highly predictive. These are complex traits, and often non-genetic factors are the most important aspect. And again, all of these polygenic scores are based on GWAS, which, as we saw, have been heavily skewed towards individuals of European ancestries. And we've seen that these scores don't always transfer too well to other ancestries and populations, and as these scores might get incorporated into the clinic, we sort of have to be wary that we don't inadvertently exacerbate health disparities. But this is a really kind of hot area of research at the minute to improve this. So finally, just like to make some acknowledgements. So in general, Melanie and all the Barlow Lab members, our Phi DivCos, Natalie and Hugh, and the seminar series organisers, Haloum, Andrew and Nadia, and then all of our collaborators on the retinal thickness GWAS here. And happy to take questions.
Should have got this earlier. All right, thank you, Vicki. The floor is now open for any questions, if you have any. I will also be checking online. Okay. So while you, there's one there. Oh, right, there's also chocolate if you ask a question.
Hi, thank you for your talk. It's not really a question, more of could you explain a little bit more? So when you had those graphs, I think one of them was about Fox gene or something, and the blue was in the corner. Could you explain, and if you could get the graph up, maybe how you read that and how that works a little bit more? Thank you.
Almost there.
Yeah,
this one.
Yeah.
Yes, so basically this is the results for 29,000 pixels, and they're all plotted spatially here. So on the left-hand side gives for each pixel the beta from the GWAS. So this is the effect size. So I'm not sure which allele it is here. So the minor allele. Each copy of the minor allele is associated with a thickening of the retina basically in this dark bluey purple region here. And the strength of this association is sort of represented on the right-hand plot, which is the p-value, but it's a log 10 transformation. So the higher the minus log 10 p-value, the smaller the p-value is. So again, you can see that the signal is sort of strongest for pixels in this region.
So what do you mean by pixels? Sorry, can you explain?
Yeah, so the thickness is basically, there's a grid of measurements, which is 256 by 120 something pixels, and they're just sort of points within the retina, and they're sort of 6 by 6 millimeter square. Yeah.
Okay, so this is kind of by location.
Yes.
Yeah, okay, that makes more sense. Thank you.
Right. Okay, while you guys are thinking, I have one question or maybe two, because I'm also trying to do an EWAS, which is a different version. But with how large the sample sizes are in GWAS, is it still necessary to even calculate for power to discover certain SNPs?
Yeah, so, I mean, with the example I showed with height, which the most recent GWAS had over 5 million individuals, that's possible with something like height. Everybody has a height, easy to measure. If you're looking at a disease which is rare in the population, you're not going to have 5 million cases to look at. You will have a much smaller number. So in these sort of rarer diseases, you might still want to do power calculations. The number of cases you've managed to assemble is relatively modest for GWAS. The other thing you have to take into account with power calculations is the effect sizes that you expect to see. So as I said, these are generally speaking in GWAS quite small, and they often depend on the frequency of the SNP. So the more common a SNP, usually the smaller the effect size. So you kind of take into account what's been seen for that trait before, whether you'd expect to observe those associations in your GWAS, and that sort of thing. So, yeah, it still can be relevant.
Yep, so there's a question online from Brendan. Hi, Vicki, thanks for your talk. What methods will be the most useful for prioritizing follow-up experimental work in the future as more and more hits are found with increasing sample sizes? So, for example, 10 GWAS significant hits for glaucoma. Is it more than 100 GWAS hits for glaucoma? Like, how would you follow up, I guess?
Cool, thanks, Brendan. So the follow-up of SNPs from GWAS is improving with the generation of other data sets. So a lot of the EQTL data sets that we look at are based on bulk tissue. As single cell becomes more popular, we'll be increasingly able to look in single cell EQTL data, and that will provide more information, allow us to look at expression in a cell-specific manner instead of a tissue-specific manner. So that's sort of one example. So basically, as all of these other sort of omic data sets become available, that will all improve our SNP to gene mapping ability and allow us to better get to the bottom of, these SNPs we identify, what are they doing, in which tissues, in which cells, and how are they impacting our disease or trait.
Thanks, Vicki, for a really interesting talk. I had a question about the intergenic hits that you get from a GWAS. Have there been any examples where there's been an intergenic hit that they were able to explain, like what the biology behind it?
Probably, yes, there would have been. I can't think of one off the top of my head. But yeah, it would have largely been through this incorporation of expression data and so on. And it also helps if you sort of got other SNPs, which are sort of linked to genes in the same pathway or so on. This kind of gives a bit more confidence in our SNP to gene mapping, and you can kind of build up a picture of what's going on, sort of from looking across the genome instead of perhaps each single SNP separately.
Hi, thanks for your talk. I'm over here. So you mentioned that a problem is that many of the studies are like European-based.
Mm-hmm.
Is there any way you can like take that into account so you can still use the data? Like, is there any like modifications?
Yeah. So, as I say, results of GWAS and polygenic scores, they're not necessarily fully transferable to individuals of different ancestries, but that doesn't mean they're not entirely informative. There is still a lot of overlap. There's a particular SNP, and you will see that association across different ancestries. It's still there. But you will also get some associations which are specific to some ancestries, and they may occur because those SNPs occur at different frequencies across different ancestries, or there could be different things going on, so different environments, and these impact your SNP trait associations. So although if you do a GWAS in the European population, you can't just expect to see exactly the same then if you look in an African population. But there is, in most cases, like to be some shared genetics across the two. So you make use of it for sure, yeah.
All right, as we draw to 12, there's literally 1 more minute if anyone has one last question. There's also none online, so I guess with that, we'll just thank Vicki for her talk for this morning. Okay.